First, I want to thank the organizers for inviting me to take part in today. It's been a really inspiring day of ideas and performances, and I hope I can hold your focus for the last little bit of time that we have here. And time is a really broad and fascinating subject. It touches on a lot of different areas. Physicists and philosophers theorize about what time is. Time is a coordinate. It's a measure of duration. It's the ever-existing entropy in the universe. Neuroscientists and psychologists try to understand how we perceive time, or more generally, how the brain makes use of it. My name is Joe Paton, and I'm a neuroscientist at Champalimaud Research here in Lisbon. And I got interested in time for a particular reason. And that is that what I ultimately want to understand is how the brain produces something we call adaptive behavior. How do we and other animals learn through experience to behave in ways that are good for us? And it turns out that time plays a role in that ability. And to give you a sense of that, I want to start with a visual analogy. If you look at this image, it's a scene, right? It happens to be of Native Americans who are hunting deer. Our ability to perceive this scene and describe it, of course, depends on the particular structure of luminance, and if this were a colored image, color in space. If we were to remove that structure by, say, shuffling the location of all the pixels in this image, of course it would be impossible to tell what this is an image of. Now likewise, if we were to remove the influence of time on our experience, we might have a similarly unintelligible mess. For instance, all of our experiences might be compressed into one moment, right? Which is reflected in this famous quote that's often attributed to the physicist John Wheeler, but what he actually read on the wall of a bathroom somewhere, which says that time is nature's way to keep everything from happening at once. But I think about time a little bit differently, because events have order, they have a direction, and there's some smoothness to how they evolve, right? So if we were to remove time from our experience, I imagine that events would become jumbled, untethered from the surrounding moments, kind of like shuffling the frames of a movie that constitutes your entire life. Now imagine trying to take that and using it to learn about the consequences of your actions in order to learn how to behave adaptively. It's impossible. Or more basically, imagine being a young child. You know, when you come into this world, you're a blank slate, a tabula rasa. And you have to learn, in large part, due to the relationships between events and time, how to really understand anything about the world. In addition, to behave adaptively, we need to have a sense of in what moment to act. So time is really fundamental in that it provides the structured order to our experience that we need in order to understand the world and make predictions, and also because we need it to behave at the right time. So when I was setting up my research group, I was super excited because I felt like I had identified this problem, one of these problems that my lab would focus on. How does the brain deal with time? Which was super important for understanding how we and other animals learn to interact with their environment. And very few people were studying it. There's not a lot known about it. And so I thought, great, I have job security for the next few decades. But I kept running up against this kind of uncomfortable reality, and that is that we're pretty terrible at timing stuff. Exhibit A, clocks. This happens to be a clock made by our 2-year-old on Father's Day, but the fact that humanity created this machine and all of its predecessors, sundials, hourglasses, water clocks, astrolabes, and all the different ways that we've invented machines to tell time, is the existence proof that our innate sense of time isn't very reliable. If it were, we wouldn't need clocks to tell us what time it is. So time can, stand still, it can fly, it can disappear, based on a number of different factors. For example, I'm going to show you 2 video clips, okay, of flashing colored grids. And I want you to look straight at the center of the video clips, and I want you to tell me which one lasted longer. And don't cheat, don't count, don't look at your watch or anything like that. Just give me your gut impression. Okay, here we go. That was the first. Now it's the second. Okay, so everyone who thought that the first clip lasted longer, raise your hand. And everyone thought that the second clip lasted longer, raise your hand. Awesome, that worked perfectly. Okay, so the vast majority of you thought that the first clip lasted longer, but in fact, they're identical in duration. Okay. And that's because, and a lot of studies have shown this, the rate at which sensory stimuli change can alter your perception of time. Okay? And here I'm showing data from one of those studies. So what's plotted along the vertical axis here is the relative estimated duration of a stimulus that has dynamics. It's changing, it's flickering, like those stimuli I showed you before. And along the horizontal axis is the speed of that change. How fast is the stimulus flickering? And what you can see is as the stimulus changes faster and faster, people show a tendency to overestimate duration. Okay? So sensory change is one factor that can affect your perception of time, but there are others. And what I want to suggest to you today, and this is sort of the central idea, is that this variability in our innate timekeeping isn't some shortcoming. It's not a problem that evolution hasn't gotten around to fixing yet, but rather it's a feature that ultimately serves our best interests. And I think it's telling that when we look into the brain, we find that those same brain systems that seem to be critical for guiding adaptive behavior also seem to be responsible for variability in time perception. And the brain system I'll spend a lot of time focusing on today is that of dopamine neurons in the midbrain. So here I'm showing a side or a sagittal view of the human brain with one side cut away. And the dopamine neurons I'll focus on today are located in the midbrain. There's cell bodies here, and this branching portion here represents the projections, or the axons, where the signals that these cells are carrying are being projected to. And these neurons, they use a neurotransmitter. They release a neurotransmitter, neuromodulator, dopamine, which alters the way recipient neurons respond to other inputs. And it can do it in the current moment, or it can alter in more permanent ways the strength of the connections between neurons, okay? Something we call synaptic plasticity. And now it's really interesting when you consider the way these neurons can kind of change or teach the strength of connections between neurons, when you consider that these cells carry a really, really useful instructive signal. They seem to teach the brain to choose those behaviors that are going to result in reward. Okay? And they act sort of like a teacher that tells you only if you're surprised at whether things are better or worse than expected. Now, why is a teacher based on surprise useful? Well, if you think about it, if you're surprised by some particular outcome, that's an indication that you should update your expectations. You should learn something so that the next time you encounter that situation, you're less surprised. Okay? These updated expectations about the quality of the current situation can be used to guide your behavior towards those situations of the highest quality. Okay, but why am I telling you all this about dopamine neurons and learning and rewards? I thought this was a talk about time. Well, one of the reasons is that these neurons project very, very densely to another part of the brain called the striatum, which you can see here in pink, which the work of many labs, including mine, has shown to be critical for keeping time. So if we can bring down the lights a little bit, this would be a little bit more clear, but what I'm showing you is a slice now through a mouse brain. What we've done is we've labeled dopamine neurons on one side of the brain with a protein that fluoresces red. And what you're actually looking at here are the axon terminals, the projections of these nerve cells. And basically everything you're seeing here in red is this brain area, the striatum. So you can just see how densely these neurons project to this area as opposed to other areas where they do project, like these ones out here. And now synaptic plasticity, remember those changes in strength in the connections between neurons, in this particular area is thought to be important for shaping the actions we take in a given situation. And if you were to look at the activity of single neurons in this area during behavior, you might see something like the following. So here I'm visualizing real data. Each circle here represents an individual neuron in this brain area, the striatum, and the brightness of the red color indicates how electrically active that cell is. And if you look at the activity of these cells, you see there's a diversity of responding, okay? And this diversity can actually be used to read out the passage of time. And this becomes more clear if we just reorder the cells by when they're active. And if you do that, you see this kind of wave of activity that moves over the population. And just like the hands of a clock move across its face, so does this wave of activity across this population of neurons. Okay, so you can read out time from these cells here, but what's the evidence that the brain is actually doing that? Well, to get at that, you need to ask the animal, this happens to be a rat where we recorded this activity, about its sense of time. If we were working with a person, we could ask them to describe their sensation using language. But we as a society don't like to insert electrodes or other devices into the human brain unless we absolutely have to. You might hit a blood vessel or you might cause an infection. However, we're a little more tolerant of doing this in an animal model organism like a rat or a mouse. But how do you ask a rat about its time perception? Well, one way to do that is to train them to make a judgment. Okay? Was the interval you just experienced longer or shorter than 1.5 seconds? And you train them until they're really, really good at this judgment. And here I'm showing behavioral data from a rat that's been trained to do this, where what I'm plotting on the vertical axis here is the probability that they categorize an interval as long. They judge it as longer than 1.5 seconds against the different intervals that we present to them. And what you can see is that for these intervals that are near 1.5 seconds, the difficult ones, their choices are variable. Sometimes they're going to judge that this interval is longer than 1.5 seconds, and sometimes they're going to judge it as shorter. And we think that most of this variability in their judgments is actually due to variability in their sense of time. So then we can compare the judgments that animals make to either observations or manipulations of neural activity that we make in the laboratory in order to try and identify candidate neural signals for timing. So the first piece of evidence that those waves of activity I showed you are actually involved in keeping time is that if you inject a drug into the striatum that just perturbs activity a little bit, it doesn't mess up the animal's behavior very much at all while they're performing this task, they get worse. So here I'm showing in orange data collected on days when we injected this drug into the striatum. Okay? And in green I'm showing data on alternate days when we just injected saline or salt water. So you can see, just by messing up activity a little bit in this area, you produce a marked decrease in the sensitivity of animals' judgments to time. But what about those waves of activity? If the animal's actually looking at where that wave is and using it to tell how much time it's passed, if that wave goes a little faster, they should think time is passing faster. And if that wave is going slower, they should think that time is passing slower. And indeed, if we record from populations of neurons during this task, we see something very similar. So when that wave goes more quickly, as you can see in this red curve, animals show a systematic tendency to judge intervals as being long, particularly for those intervals that are near the decision boundary. And the reverse is true when that wave progresses more slowly. Okay, so you have this clock, and it's variable. Sometimes it goes faster, sometimes it goes slower, and that predicts the judgments that animals are going to make. But what's responsible for that variability in clock speed? And this is where we come back to dopamine. So if you look through the psychological literature and you try and find situations where your timekeeping seems to be systematically altered, on the right what I'm showing are cases where time appears to drag. And this can get confusing because this is sort of akin to having a fast internal clock. You think that more time has passed than actually has. And these are things like fear, anxiety, boredom, and pain. And on the right, I'm going to show cases where time appears to fly. And again, this is akin to having a slow internal clock. You think that less time has passed than actually has. And these are things like your motivation to approach or engage with a situation, attentional load, pleasure, and novelty. Now, interestingly, most of these cases here on the left are associated with decreased dopamine signaling. And most of these situations here on the right are associated with increased dopamine signaling. In addition, conditions where the dopaminergic system is damaged, such as Parkinson's disease, people with Parkinson's disease display kind of peculiar timing deficits. So there's a lot of kind of indirect evidence that dopamine neurons may be exerting some kind of control over your perception of time. But how do we directly test this idea? So recently, Bassam Attallah and Sofia Suarez in the lab devised and carried out a series of experiments to directly test whether dopamine neurons can control your time perception, or at least the time perception of mice. So what they did was they first trained mice to perform this judgment. Was the interval longer or shorter than 1.5 seconds that you just experienced? Remember the data I plotted before, which is showing the probability that an animal makes a long judgment as a function of the different intervals, because that'll be important. And next what they did was they measured the activity of dopamine neurons using an optical technique, where we engineered dopamine neurons to light up when they were active. This gave us a fast and continuous measure of the activity of these cells that we could then compare to the judgments that animals were making from trial to trial. And what they observed was striking. When the activity of these neurons was higher, as you can see in this blue curve, animals' clocks appeared to slow down. They were more likely to underestimate duration. And the reverse was true when these cells were less active. They were more likely to overestimate duration, as if their clock had sped up. Okay, so there was this correlation between the activity of these neurons and the pattern, the judgments about time that animals were making. But of course, correlation doesn't imply causation. So next they went a step further, and they applied a technique from a relatively new field called optogenetics. The name should make sense in a moment. In order to transiently manipulate the activity of these cells during this behavior. So what is optogenetics? In our eyes, we have proteins that couple to light-sensitive molecules. If these molecules absorb a photon, they change shape, and this kicks off a series of steps that ultimately results in a message being sent to your brain, and that's how we see. But this process is kind of complicated. It involves a number of different proteins and genes, and in the end of the 90s and the early 2000s, people were wondering if they could kind of harness that general idea to make neurons controllable with light. And what these people did is they identified another similar kind of protein that's expressed by algae that actually is light sensitive, but it couples directly to an ion channel, making it easier to kind of express potentially in neurons. And they managed to express these things in neurons, rendering them controllable with light. And this has brought about a real revolution in neuroscience because it allows us to go beyond just identifying correlations between signals we observe in the brain and particular behaviors we're interested in or particular brain functions, and actually inject in signals into particular genetically targeted cells during behavior. And so that's what these guys did in their experiments. They induced dopamine neurons to express these light-sensitive opsins, and then they inserted a little glass fiber right above the dopamine neurons. And then randomly throughout these behavioral sessions, they would illuminate these cells to either activate or inhibit their activity. And what they found was indeed, when they activated dopamine neurons, they were able to apparently slow animals' timekeeping, consistent with the observations they had made in the previous experiments. And conversely, when they inhibited these neurons, though the effect was a little bit more modest, they caused the animal to apparently speed up its timekeeping. They showed a tendency to overestimate duration. So dopamine neuron activation slows, and inhibition seems to speed timekeeping. And so I want to step back now and ask a more general question, which is why? Why would this brain system that seems so important for teaching us how to act in order to get rewards also be able to influence our sense of time? And this is the crux of the point. And that is that a big component of your sense of time is actually how worthwhile your current course of action is. Okay? The quality of the current moment is actually warping your sense of time in order to keep you in pursuit of those situations that are better than expected, or to cause you to disengage and potentially move on to some more rewarding situation if things are less than expected. And there are a lot of situations like this in life, right? Life is sequential. We're always posed with the question, should I keep doing what I'm doing now, or should I switch and do something that could potentially be more rewarding? Take, for example, you're on a first date. It's going horribly. You're thinking about all the other things you'd rather be doing as each interminable second is creeping by. So you skip dessert, you leave early, and you're off to greener pastures sooner. Well, let's imagine another scenario. The date's going really well. You're completely focused on the current moment. And before you know it, you've been there for hours, night falls, and then the sun rises. And during this entire time, you form this deep bond with this person that just might form the basis of a lifelong relationship and all of its associated benefits. So the next time you notice a discrepancy between your inner clock and the watch that you're wearing or the clock on your cell phone, think about those dopamine neurons in your midbrain and how they might be speeding up or slowing down those waves of neural activity in the striatum, pushing around your time perception. And all the work that evolution has gone to keep you in pursuit of things that are good for you. Thank you.